Welcome everyone to the August 19th IMAG MSM Working Group on Multi-Scale Modeling and Viral Pandemics meeting. We have two great mini seminars lined up for today. I need to remind you that the meeting is being live streamed on YouTube and is recorded. So please be aware of that. As always, Reinhardt and myself are the co-leads of the working group. Bruce Shapiro, who you've had correspondence with, is on the call. Jim Sluka, whom you've also had correspondence with, is away today. And you can reach us at your convenience if there are any suggestions, comments, or questions that you'd like to have. As always, also, we have our various media channels. They work better if people use them. Please help us out with them. Especially the IMAG Wiki page could use some love. And we do have an IMAG LinkedIn page. That's a good opportunity to share not only the IMAG work, but any work that you've done that's related to the topics of the meeting. Please feel free to share it there. And we have our YouTube channels, which we'd like to publicize as well. Open opportunity for any short announcements before we go further. Okay, there are a couple of requested announcements. We were asked to remind people that there is a virtual workshop on virus dynamics coming up in October out of Seattle. Fred Hutchinson Cancer Research Center is organizing it. It looks really good. They have a deadline of August 31st for submission of abstracts. And so we encourage people in the group to submit to that. We've been asked to try to drum up attendance. It sounds like a great meeting. I know I'm going to be submitting an abstract. If anybody has any specific comments about that meeting that they'd like to add, please go ahead. I have a request, which is, I mentioned the YouTube channels. I and Giuliano have been rough editing the videos and annotating them and putting them up on YouTube in our YouTube channels. But they could definitely use some proofing and some attending. Two, if anybody is willing to volunteer to review not the details, but simply the splash pages and the content of the annotations, I would be most grateful for the assistance. So I'm reaching out to people. If you're willing to just do one or two, if everybody took one or two and cleaned them up, we would have much better access. So please contact me if you're willing to do that. The next thing I wanted to talk about, which is also an announcement, we have a steering committee meeting tomorrow, which will be discussing this, is that our working group is almost a year old. And it seems time to be evaluating what's worked and what hasn't, and what our plans should be for next year. There is a document with a link shared in the chat to which people could contribute. It has some possible questions. What is the role of the working group going forward? What should the working group be doing? What could we do in terms of expertise and community building? We have a lot of people at various levels of engagement with a lot of expertise. Is there some collective set of projects that we could use to leverage this collection of expertise? And similarly, are there projects that would bring in new people or engage the people that we have more intensively? Is there some expertise that's missing from our current membership that we could be seeking out? And then in terms of scientific aims, over the past few months, we've talked increasingly about the concept of an immune digital twin as a potential goal for many people. Is that appropriate? Are there other aims that would match our expertise sets better or would expand our community or be more responsive to the existing community? Are there other aims that we should be having? I don't expect answers today, but for people in the steering committee, please think about those. And then next time, we're going to have an open forum for discussing these topics. So please think about them and be ready. So again, the link for that is in the chat. There's a Google Doc. Should have access to that. So next week, we're going to have a future of the working group discussion rather than seminars. I hope people will participate. We'd really love to have your feedback. Reinhard, do you have anything you want to add to that?
No, you did a great job summing that up. I think it's really important, and I hope that many people will attend, and before that, we'll make contributions to that document.
Okay. So next week, we have the future planning meeting, open planning meeting. Then September 2nd, we have Harry Hochheiser from Midas speaking to us, Guido Espana from Notre Dame. We have one open slot left on September 9th. September 16th, we have the head of applied biomass speaking to us. September 23rd, we have the very distinguished Russ Taylor from Johns Hopkins, who's done a lot of work on digital twins and automation of medicine on very large scales. I hope that didn't seem to imply that the other speakers aren't highly distinguished, but Russ is a bit of a coming in from a very different perspective. And so I think that will be really interesting. We have opportunities for somebody to speak on September 9th. Please feel free to volunteer yourself or one of your postdocs, or if you're leading a subgroup, to present about what you've been working on or what you think you should be working on. So please send your suggestions to me, Reinhard, or Bruce. Please try to remember to mute your mics and hold your questions until we're done with the short talks. Again, we know it's hard for our speakers to get everything in 20 minutes. We appreciate you summarizing things. If you don't have enough time to say everything today, we hope you'll come back and speak to us again soon. With that, we know that we don't do elaborate introductions here, so I don't want to take any more of Dean's time. Really appreciate his being willing to come and speak to us today.
Thank you, everyone. Thank you, Professor Glazier, for inviting me. And let me share my screen here. I'm not 100% sure how you got my name, to be honest, because I never told anyone outside of Takeda that I was working on anything with viral dynamics. But I'm glad you did, and I'm happy for the invitation. Okay, let me switch. Okay. Does anyone know how to make the little bar go away on the top? Hide floating meeting controls. Okay, great. Can everyone see my screen? Great. All right, so this was a group effort, and obviously it started with COVID, and very shortly after our first lockdown, we were asked to evaluate different oncology compounds for their potential in treating COVID-19. And the working group was that we quickly put together was myself, Hojat Bazazi, Hailim Cho, John Gibbs, and Eric Fedek, with key guidance from Kathy Terlau-Stewart, Ajita Dash, and Majid Vakil-Najad. And the academic partners that generated the in vitro data, let me get my pointer going here, were the Michael Gale Lab at University of Washington, Ralph Barrick Lab at University of North Carolina, Tim Spicer at Scripps, and Matthew Freeman at University of Maryland, and their lab members. So the main question we were asked is, can Takeda drugs be used to treat COVID-19, SARS-CoV-2 infection? And given that we're planning on collecting, or had these academic partners generating in vitro data of anti-SARS-CoV-2 activity of our compounds and cytotoxic activity as well of our compounds. And then the question would be, how do we translate the in vitro activity to clinical activity, and taking into account differences in unbound fraction of drug between human blood and the medium that the in vitro studies were performed, and can we attain those active concentrations in the clinic in patients, given that there are obviously toxicity and other limitations in how much concentration you can attain in patients. Then there's also understanding the difference between in vitro viral load assays like reporter fluorescence versus in-host viral load measurements, for example, PCR of swabs taken in the nose or the throat. Then there's another question about establishing a relationship, if any, between in-host viral load and clinical benefit. I unfortunately won't address all these questions, but I'll try to speak to all of them at least. So part one is the in vitro translation of the in vitro data in a clinical PK pharmacokinetic context. Sorry. So again, the question is, can we attain the in vitro levels of Takeda drugs associated with anti-SARS-2 activity in actual patients and humans, given the pharmacokinetics, so the drug concentration time course in the blood at approved and tolerable doses and schedules, and the differences in protein binding of the drug to in vitro medium versus human plasma, and as well as including the in vitro cytotoxic effects that we don't want to see. So the basic methods that we set out to perform is to fit IC50 curves, very simple Hill type equations, to the in vitro data. And then the in vitro data is both percent of infected cells as a function of drug concentration and percent viable uninfected cells relative to control as a function of drug concentration. Then we use what we know about the pharmacokinetics of each drug. Most of them were out on the market already, and so we know what the approved, or if they're not approved yet, what the maximum tolerated dose is for these drugs. And then we estimate the pharmacokinetic time course of concentration in actual patients and the variability among patients in that. Then we use some equations that one of our collaborators at Takeda, Hoja Bazazi, made the derivation for converting from in vitro unbound fraction to in vivo unbound fraction, so that we're comparing apples to apples in terms of the relative concentrations. And then we overlay the clinical pharmacokinetics on the in vitro concentration effects curves, and then identify the compounds that have selective antiviral activity that's clinically attainable. In the future, in the following slides, rather than get permission from all 9 project teams, that we just did not have time to do, given that this data has not been shared officially outside of Takeda, we had to blind the names of the Takeda compounds. So they're color-coded, but the ones we could unblind were the controls. So we have remdesivir, which is an IV antiviral, originally against hepatitis C. It's been approved more recently because it shortens hospital stays in patients with COVID-19. Hydroxychloroquine is famous and infamous for various reasons, was originally malaria, and some have suggested potential effects against SARS-CoV-2 and COVID-19. And then pyromycin is an antibiotic that was used only by one lab as a sort of a control compound, or a comparator. Sorry, some of these slides animate by themselves for reasons I don't fully understand. And so the lab one, we used Calu-3 cells and 4% fetal bovine serum, run for 48 hours, and they used a fluorescence assay as a reporter of one of the viral genes to measure viral load in the cell culture. And the main point of this slide isn't to have any take-home messages, because the point of the slide is that we found it hard to take home any messages from overlaying all these compounds on top of each other at the same time. Of course, we could have flipped through, tens of slides, but we wanted a way to understand how the compounds were doing relative to each other at a glance. So what we did was oh, sorry, yeah, this is this free fraction concentration calculation. So what we did to address that is to try to plot each compound in a two-dimensional space where the x-axis is percent cytotoxicity relative to control, the y-axis is percent antiviral activity relative to control, and then the curve is parameterized by concentration through this space of where the antiviral activity and cytotoxicity take you as you increase the concentration. So this is a very boring compound that linearly increases both, or proportionally in equal proportions, for example, both cytotoxicity and antiviral, just for the purpose of the schematic. And so the coloring of the curve is parameterizing the concentration of the drug. The dashes mean that we're in an extrapolated range beyond the support of the in vitro data. And then this little fat sausage type thing around the part of the curve gives you the minimum concentration at steady state and the maximum concentration attainable in humans, in the typical human. And then the average concentration from a typical human is given by this asterisk here. So that marks that concentration. So this is supposed to be a way that at a glance you could look at several compounds and decide which one of them are promising in terms of doing what we want them to do, which would deplete virus first, so go upward on this graph, and then bend towards the right, maybe at higher concentrations, at superclinical concentrations. What we don't want to see is a drug that kills cells first and then starts depleting viruses at higher concentrations. So when we did this to the Takeda, sorry about that, to the Takeda compounds, we could see, for example, that our positive control remdesivir did look good by that criteria. It robustly depletes the virus up to 100% before it starts bending off and causing anticellular toxicity. And so that said, something to keep in mind about remdesivir is the average concentration is way down here somewhere. You can't even see where the asterisk is for remdesivir. But remdesivir, you have to keep in mind, is a prodrug, and it's the nucleoside product inside cells that is really doing the work. So it's not necessarily fair to remdesivir to include to evaluate it in terms of its absolute blood concentrations. But nonetheless, it does look good by this, at least by its shape. Unfortunately, none of the Takeda drugs had similar shapes, and not surprisingly, they're oncology drugs, so they're pretty good at killing cells, which is not exactly what you want when you're designing an antiviral drug. Some of them did show at least the right concavity, like this purple one here, but not approaching that of remdesivir. Hydroxychloroquine, as another control, performed similar to remdesivir with its concentrations, attainable concentrations, predicted to provide about 50% inhibition of viruses, and then the bending off would be towards toxicity, was predicted by this approach to happen at superclinical concentrations. When you add well, we'll talk about that later. Puromycin showed a curve here. There were some Takeda compounds that had some promise based on these graphs in terms of their predicted PK and killing viruses before they start killing cells. But a lot of them ended up in this not so great region here. Another way to summarize all this data in a sort of a tabular format is to, for any given drug, we have the horizontal axis is the nanograms per milliliter free fraction corrected human concentration. And for each of the 4 or 5 labs, so I only showed 2 of the labs, but there were, I think, 5 labs that we got data from. There's a range of the in vitro IC50 to the in vitro IC90. And if there are empty circles, that means toxicity is unknown, for some reason wasn't assayed or measured, in that assay. If the tox is greater than 20%, they're color-coded red. if their tox is moderate, so 10 to 20%, they're yellow. And then if they're less than 10%, they're color-coded green. On the same axes is overlay the minimum concentration, the average concentration, and the maximum concentration for a typical patient for that drug. And then we calculate using the best lab. So the best case scenario is the drug acts like it does in the lab that has the furthest left IC50 value, for which the toxicity is yellow or better. So you take the best lab for which the toxicity is yellow or better, and take the ratio between that and the average concentration. We call that rho. And so this was a drug with a less favorable rho, greater than 1, and then a more favorable drug would be one whose rho was less than 1. That's about it. So when we did this and then ranked by rho, surprisingly, I don't know if this is surprising to everyone else, but hydroxychloroquine actually bubbled up to the top in terms of selective antiviral activity according to the in vitro assays, then followed by a couple Takeda compounds. But then the ratio flipped from negative to positive, or sorry, below 1 to above 1 between these two compounds. And then remdesivir, for I think reasons I alluded to earlier, actually came down 4th on the list. And that's probably because, again, it's a prodrug, and so its concentrations aren't necessarily predictive of its mechanism of action and its activity. So then there's all the other Takeda drugs that had, high to extremely high to not even accessible rho values because there were no safe concentrations according to the in vitro assays for the cells involved. So that's part 1. Do you prefer to go all the way through and then take questions? How does this work?
Well, we have about 5 to 6 minutes, 7 minutes left here.
Okay.
So,
all right, I'll just go on to part 2 then. Okay. So part 2 is the in-host viral load dynamics. And there were several models to choose from at the time. There's this classical Parelson model that has the virus and target cells interacting in this manner here. Around the time we started this, Rada Savage came out with a preprint where she had a model that had the drug effect killing virus at a rate proportional to the viral load. And then you had an immune effect growing up exponentially without limitation. We came up with a very similar model that was had an immune effect, a very simple, obviously not non-mechanistic immune effect that had an antiviral effect that increased with time at a certain half-life with a certain half maximal activation time. And we came up with this model and fitted it to about 100 patients worth of data. That was all we could find at the time. This was early on in the pandemic from the German study, a French study of hydroxychloroquine and azithromycin, and a data set from China and from Singapore. And later on, we actually, there was the Royal Princess or some, I can't forget the name of the ship, some sort of royal sounding prince is kind of ship where everyone got COVID. And that was another data set that we used. So anyway, the very simple model, this just, I didn't have time to show you the goodness of fit, but it met most of the criteria for model fitting to data. So very quickly, I'll show you what I did was built this little app to help us build intuition about this. This was actually done before the in vitro data came because we had to move at risk with a modeling project and have this ready to go in case some of those compounds ended up being more promising. So just very quickly, it's just a little app. How do we get out of here? So all this lets you do is try out different scenarios for when you start the drug. So this is consistent with the idea that if your drug effect starts earlier, you can avoid and reach negativity much faster, which isn't particularly surprising, but worth quantifying, I think. Now, drug effect on viral clearance turns out to be, of course, the key parameter. If you crank that up, you get faster killing and so on. According to this model, if you modulate the immune system, the model predicts slightly faster resolution of the infection. Speeding up the immune system doesn't seem to do that as much over a large range. So, and then it comes with a model description and so on. So I built this in anticipation of one of the drugs being, well, in anticipation of several things aligning. But going back to my presentation. Okay, so yeah, so we did that in vitro free fraction correction, combined it with pharmacokinetic knowledge of those compounds, and we saw whether Takeda oncology compounds were potentially active against COVID-19 in the clinic. We concluded that there were I think there was one that was promising that we started following up with the project team. Then before that, we made an in-house viral load dynamics model using about 100 patients with SARS-CoV-2 PCR data. And the ultimate linking, the reason why we built both of these models, wasn't done because, for starters, the unfavorable in vivo projections caused the entire effort to be deprioritized in favor of what I call my day job, which is modeling oncology. And at the time, it was very hard to find data that established any relationship between viral load resolution and avoiding hospitalization or mortality. And in fact, the paradigm at the time, and this is March 2020, was more along the lines of it was the cytokine storm killing the patients more so than the virus. And so we decided, even if we could find the data, we didn't have the data. If we did, we weren't sure it would show us much of a link between viral load and mortality. Oh, I wanted to end with a teaser. Let me hide this thing. So my colleague, Andy Zhu at Takeda, did a very nice modeling effort, more mechanistic systems type modeling of the bradykinin pathway, kallikrein kinase system, to evaluate one of our non-oncology drugs that modulates bradykinin levels. And that was a successful modeling effort that was completed and helped drive internal decisions on pursuing our internal drug for COVID-19. So I noticed an empty slot. Anyway, if you're interested, I'll put you in touch with Andy. And I think that's it for me. Yep. Are there any questions or comments?
Okay, thank you very much, Dean. Normally we go through both talks before we have questions.
Okay, thanks.
But again, if you're able to stay.
Yeah, I can.
Great, thank you.
Okay.
I know it's a slightly unusual format, so I appreciate your patience.
No, that's fine. It's more like a panel, right? So.
Okay, so our second talk is from Juilee, talking about discrete state modeling of human immune response. And I don't want to take any more of your time, so let me turn it over to you.
Great, thank you. It's my pleasure to give a talk. Wait, let me see, I can't. Yeah, it's my pleasure to talk about our research and the subgroup that we haven't really started. Let me first share my screen. Yeah, so many things are open, so couldn't find my presentation, but now here it is. So you see it, hopefully, everybody, okay. So yeah, so it's, there was So yeah, it's pleasure to be here, talk about the discrete state modeling, which my lab mostly works on, along with many other things that we do. And I wanted to connect that with the subgroup that I was supposed to co-lead, but it didn't really get traction. The subgroup was knowledge acquisition and modeling to investigate human immune responses. So I think this talk hopefully can drive some of the questions and thoughts about what should be included in this subgroup. I initially tried to get the group together, but I think the areas of what knowledge actually means was so different among the co-leads, and at least this is my interpretation, that we didn't really get together in a coherent fashion. So I think this talk and the questions from all of you will really help us direct some of the thoughts. Yeah, so with that, we mostly work on human immune responses, and the human immune responses are very challenging to address, and some of the challenges are that it's very diverse response and are coming from very wide range of cell types in distinct states and differentiation and activation. There are new technologies, including single-cell RNA-seq cytokines, which really allow us to monitor what's going on in various diseases, infections, using these technologies. However, mechanistic inference of these observational data is hard, since we cannot really do perturbation analysis in humans. So I think that really drives some of the research ongoing in my group. We try to look at these human responses and think about the variation and the clinical and other covariates that can drive some of this variation, and best way to analyze data. And some of the variation, as we all know, is caused by immune history, number of infections we have had, gender, age, and it's not always straightforward to optimize these responses against these different populations. And these are some of the challenges that we are trying to work on in my group. And the way we try to handle this is using mechanistic models, mechanistic computational models, and developing these models and integrating with knowledge and data curation. And this is really where my interest in this group, the subgroup or working group came into. So I should also say that I think I missed some of the talks in this series. So if I had missed any talk related to this topic, please just raise your hand and let me know, or we can discuss that at the end of the seminar. So again, going back to our approach is to combine this knowledge that is existing in the literature and the public domain, and combining it with computational modeling. And we frequently work with human cohorts, where we recruit cohort and are involved in the recruitment, designing experiments, high-throughput experiments, and characterization of these responses. So I'm not going to go in the detail of the methods that we use, but just want to mention that we use plethora of the methods, going from abstract empirical analysis, which only is based on the data and do not incorporate any prior assumptions, and going right on this bar, we try to incorporate more and more prior knowledge and try to develop mechanistic models. And particularly, my group is interested in developing discrete state models because of its simplicity and the fact that we don't really need to estimate a lot of parameters. But again, of course, there are other caveats and pros and cons of using these different methods. Just to quickly introduce what I mean by the discrete state model is that we basically start with the network, which can be represented like this, which is mostly the ones that we are considering are the directional networks. And then there are logic gates that are describing the regulation by different edges or upstream nodes. And this is given by a Boolean function over here. And then we can simulate these update rules using using different simulation methods, such as asynchronous update or synchronous update. And again, there are many different ways of doing that. We have also tried using fuzzy models where we don't only have two states, but we can incorporate multiple states to model these networks. But again, I'm keeping it short for the in benefit of the time that I have. So what my group has done recently is the way we wanted to think about this discrete state model is that we wanted to apply these models to the large scale omics data. And to be able to do that, we use this pathway analysis framework, which is very common, as many of you might know, in analyzing and looking at the omics data. So frequently frequently used methods, what they do is they use this high throughput data and only use sets of genes and not really a network topology, right? So using these sets of genes that are identified from the data, you overlap with those with the known information, and you can we can do pathway analysis to find out the functionality of the genes that are responding to the treatment or infection. So there is this knowledge curation in this as well, because now we are using many different types of gene sets, right? So there are many groups who are actually working on optimizing and improving this characterization of gene sets, which we can use in this pathway analysis. But what my group is mostly looking at, we are thinking about networks and the topology of the pathways. So we are interested in not just the sets of genes, but in thinking about the topological characteristics of the network and combining that with the high throughput data, and using that then to perform the pathway analysis and identify the pathways that are significantly different in the groups of comparison across, again, different infections and diseases. So that's exactly what we have done. We have these 2 years back, we published a method called Bonita, which basically will use genetic algorithm along with local search to infer the rules and infer the rules from the high throughput data using the pathway topologies. And then we use this to perform perturbation analysis. So basically we perturb the nodes in the pathways and then identify the state of the network and look at the difference between the state of the network to score the pathways that are most differentially regulated. So this paper is published and we compared our Bonita-based pathway analysis method with existing gene set analysis or the pathway analysis methods, such as Camera, which is one of the most popular methods, but it does not use pathway topologies, and Clipper, which uses pathway topology. And when comparing with both methods, our method did very well at the reduced signal, but all the methods did comparably well for when the signal was very strong. So this signal, the way we are thinking about it, it could be TLR activation or activation of any pathway. So any signal, when you think about the pathway, and that's the signal that is plotted on the x-axis here, and on the y-axis is the significance identified by these different methods. So I'm not going to talk a lot about the applications. We have applied this to several data sets. Here is one nice application, just want to highlight. In this case, we have clinical trial data for after HIV vaccination, and we were interested in finding out that the activation of the B cell receptor signaling after two types of vaccines. One was, so basically the two vaccines had different vectors, and we wanted to ask question if there is any difference in the BCR activation. This is what I'm showing you. Basically, this model was, so in this case, this is a logic-based discrete state model. As you can see, the black nodes indicate AND gate, and whenever there are no black nodes, that's OR gate. And this study was identified some of the genes which we validated in vitro systems. So this was a great success for Bonita application. And now we are extending that Bonita application to the single cell RNA sequencing data, which is really a great type of data which allows us to connect molecular networks with the cellular phenotypes. And we can now connect the attractors of this model with the cellular phenotypes and try to identify how different the cells are using single cell data. I should point out that there are some of the other methods that also use something similar for the inference of the logic gates. The key difference between those methods and our method is that this can work with the cross-sectional data, and which is more common in humans. So we frequently have cross-sectional data and do not have longitudinal data for which these other methods are meant to work with. So for the single cell data, this work is not yet published, but we again are looking at HIV positive people with and without atherosclerosis. This population is extremely well characterized. We have imaging data to characterize this population. And we developed Bonita for single cell data, and now have now can perform the pathway analysis with this data to identify pathways that are different in the people that have atherosclerosis and people do not have atherosclerosis. So this is what I'm showing you here for one of the cell cluster. So this is actually this graph shows you all CD8 positive T cell clusters from the single cell data, and the pathways that are different between atherosclerosis positive and negative people. And x-axis is basically the p-value. So many interesting pathways we identified as being differentially regulated, including one of the pathway is HIF1 alpha. And what now the single cell Bonita now allows us to really look at these pathways and think about how that affects the cell phenotypes, sort of cell phenotypes, or to characterize the cells.
Okay, just a 5-minute warning.
Okay, yeah. So here basically I'm showing you that the networks, the impact, the importance of the nodes after perturbation, and then you can see here I'm only showing you the cells that are present in AS positive and AS negative people, but for this JAK-STAT pathway, we can actually characterize the attractors of the cells and see that the cells are coming from belonging to different state of activation of this JAK-STAT pathway. And again, looking at the node size is showing you what genes are important. So yeah, basically Bonita we found very useful, and we found its application with the single cell data and RNA-seq, bulk RNA-seq data. But now I want to quickly go over and think about knowledge curation. So we can do some of these modeling techniques, but where we are finding challenging is in the knowledge curation and how we can use some of these pathways in best way for modeling. And that's really was driving some of the thoughts that I had for this group. So there are some of the ways currently used for defining the pathways, for storing the mathematical models such as SDML. So however, I have some of the questions that I think are not really addressed by using some of the methods. So the programming languages that store the pathways, such as biopacks, have a lot of metadata. And when we are thinking about the metadata, how should we use this metadata in mathematical models? And especially when I'm thinking about it, it's really automated development of the mathematical models for large-scale data set. Are networks the best way to represent the pathways, or there are other ways for representation? If we want to simplify pathways in the format of the directed networks, how do we include these metadata? So just to give you a quick example, we were trying to convert all the biopacks pathways from pathway commons to for our modeling, and it wasn't straightforward, and it wasn't straightforward to convert all the metadata in a format of the networks. So this was one of the question, and then second question is really when we are using this large-scale omics data. There is sparsity in the data, especially when you are thinking about single cell data, single cell attack seq data. Combining that together, there are genes, there are technical variation in the expression of these genes. And how do we separate that from what is known in the public domain in the databases? That was another question I think is worth addressing for this community. And again, how to standardize some of the molecular interactions. To quickly give few examples in this direction, what my group is doing. So for molecular network of antiviral responses, we try to connect by using we try to connect antiviral networks with some of the gene sets and do have a nice antiviral response network. But again, there were a lot of challenges that I just described we faced when constructing this. And for antiviral network, we have a lot of information in-house that facilitate development of this, but for some of the other models, it might not be present. So in that case, how do we standardize this construction of large-scale models? One of the other work that we have done in this direction is we recently developed Wiki Networks, which standardizes all the networks in all the pathways in the simplified network format from Wiki pathways. And I'm happy to talk more about this if anyone is interested. This paper is under review, so hopefully it will be soon out, and you can use this to convert Wiki pathways into networks. We are also trying to develop algorithms to really understand networks from large-scale data sets. So in this case, we start with a co-expression network, cell type specific co-expression network, and then construct the gene set specific subnetworks for each gene set. And this is one example of how our gene set network would look like. So all purple outlined nodes here are the nodes in the gene set, and we identify the other genes that could be involved in that network by using this algorithm. Yeah, I'm sorry, it was very quick. I had a lot to talk about, and some of the things that I spoke about was very quick, and I'm happy to elaborate on any of those topics. I want to thank all my group members. The antiviral network was developed by Raymond. Wiki Networks is developed by Mukta. Bonita is developed by Rohit, and some of my collaborators. And thanks to my funding.
Okay, thank you very much to both of our speakers. Would somebody like to lead off with a question for either one? And again, we'll stay in the room for at least till 4 o'clock, and then at 4 o'clock, depending on how people feel, we can go to breakout rooms or stay together. So please, questions for either speaker. Well, I always have questions, but I don't like to be the one who starts. Well, I guess I will be then. So I guess, Dean, let me start out with you. Why did you decide at the beginning? You mentioned at the end that you looked at things that were more immunomodulatory. Why did you decide initially to focus on antivirals as opposed to other potential drug-based therapies?
Yeah, that's a good question. Well, my scope is oncology usually, and so I was called in for the purposing of oncology specifically. But yeah, there was that other one that I did not work on that Andy Zhu did, that was more immune and immune modulatory, I think, in nature. We have a large oncology pipeline, and then sorry, there were a couple papers, like computational chemistry type papers, that showed that some of our compounds had a very good chance of binding to one of the proteases in the viral genome, and specifically our protease inhibitors, but some others as well. Some of our CML compounds as well showed a chance of doing that. So I think that's what sparked the initial interest in exploring the oncology compounds.
I guess again,
sorry, I should mention the other one is there's one among them that is an interferon stimulator, which may be a bad thing. We can't decide. We didn't know at the time. Yeah.
I guess it depends when and how it stimulates interferon activity.
Right.
Okay. Other questions?
Yeah, I have a question for Julie. So if I remember your algorithm correctly, you derive Boole networks from single time point gene expression measurements. Is that correct?
Yes.
So how do you? I mean, why is it that this gives you actually interesting dynamic information? Because Boole networks ultimately are dynamic models.
Yeah, so we can simulate the dynamic trajectory given the rules. There are, of course, caveats, because because we assume that the cross-sectional data is at the steady state, and with that assumption, we fit the models. And in that case, there are several solutions that can fit the observed state. So we actually have ensemble set of models, and we call it ERS, ensemble set. And then in some cases, when I was showing you attractor analysis for the single cell data, we sample from this ERS to find out the trajectories, and if those trajectories are different in some of the cells given the data. But we don't have data to fit that observation, that we just basically use the models that are optimized for the cross-sectional data. So yeah, we could use it and try to use the dynamic information for making some predictions, but we frequently don't have the data to fit the models. So in that case, we typically go to in vitro model systems where we can play around with few of the observations and try to test those predictions.
Thank you.
Julie, I wondered is the ensemble approach assumes all models are equally likely, and or can it take into account maybe if you have differential confidence in certain data points than others, or certain measurements than others? Could you use that to somehow have a non-uniform likelihood of each model?
Yes, and we are thinking about ways to do that. At this point, I think we can say that we have increased confidence for modeling some nodes than other nodes in one pathway, because frequently we know that few of the nodes always fit much better and the errors are much lower. So summarizing that, I wouldn't summarize that at the network level. So typically, if we have multiple models, we would choose the nodes from those networks for validation that have highest confidence.
Mm-hmm. Can you seed also in maybe in a Bayesian way arrows that you know are there based on biology before you train it to the data, so you can require them to be in the model?
We have a way to do that. Actually, we haven't done it though. We do, and currently, so there is a probabilistic model underlying in the Bonita developed for the bulk data, not for the single cell data. And we do prioritize some of the edges. So we typically will not include all the edges. We include top 3 edges that we think based on the prior knowledge.
Okay, thanks.
Other comments or questions? I guess one thing, Dean, we're very grateful to you because we've been eager to have more pharmaceutical participation in this working group.
Good.
And so one thing we might ask is, you showed sort of classic PBPK, PK modeling and scaling, and then you showed some simple Allen-Pearl-Sodian type in-host bioreplication models and clearance models. Is there anything where you feel that people doing more other kinds of modeling might have approaches that could be valuable in the world that you live in?
Yes, certainly.
Are there problems we should be working on that we might not know we should be working on?
That's the second question is harder, but no, certainly we're open to different approaches. Technically we become a quantitative systems pharmacology modeling group. It's just I use whatever techniques make sense at the time, given the data that I have in the question. And so anything that increases our bag of tools, I think, would be extremely helpful, like what Julie showed today, and also more mechanistic models where we sometimes invest in quantitative systems pharmacology models, and there are times when questions come along and we have to grab something off the shelf, so to speak, and it's usually an academic publication of a systems pharmacology model. And last time I tried to do that, it takes about a month to reproduce the paper. And so this was several years ago, so I don't know if the state of the art has changed since then, but certainly reproducibility is an important feature if you want to see your work eventually used for pharmaceutical decision making, for example. I think that would, putting it in a repository or having even a version controlled system that lets you unambiguously point to code that created this figure. I think that extremely helpful in helping us get up to speed and build confidence in a model before we start applying it to our questions.
I mean, it interested me a lot that you were able to get what you felt were very meaningful data sets using the application that you showed, using such a simple viral infection and immune response feedback model.
Mm-hmm.
So John Rice isn't on the call at the moment. Usually he'd be asking the question about how do you build confidence in the model? You mentioned you validated against the 100 or so patient data sets that were available. We've heard other people talk about how limited the availability of longitudinal data, especially for viral, even for viral load, not let alone immune state, is for patients with SARS-CoV-2. What are the sort of, how do you think about validation of the model to give you confidence in your work that it's predicting something that's useful to you?
Right. Yeah. I wish I'd included in my slides. I don't know. I have them laying around somewhere. There's a little primer on nonlinear mixed effects modeling, which is our tool of choice, usually in pharmaceuticals, because it lets you allow for inter-individual variability while fitting to the population simultaneously. And when we do that, the tendency is, and it usually comes with diagnostic tools that let you do internal checks, because rarely do we have enough data to reserve half of it, say, for a validation type approach. And validation is a tricky word anyway. When you have something trained on 50 individuals, you can't predict the individual time courses of 50 individuals the model hasn't seen before to the same extent and goodness of fit of the ones you trained on. So there are other internal checks, and there are things, like visual predictive checks, they're called, that you see how well the model captures the trends in the data and the variability as well. And so, yeah, building confidence usually just means looking at several diagnostic plots. Not all of them will be 100% perfect, across the board. And then you start looking at the trade-offs between which model has a lower likelihood score or Bayes information criteria, so penalizing for more complexity, but trying to get a better goodness of fit at the same time. And so sometimes we look at that. Anytime you look at one thing, you end up inevitably having things in the background getting worse that you forgot to look at. So the key is always to remember to look at all your diagnostics every time before you make a judgment on, there's no one size fits all for choosing the best model. And so, yeah, for my purposes, it also depends on what you plan on doing with the model. If you plan on using it for forward simulations, like I did, then you do need to make sure some of those predictive checks are working out right. If you're just trying to estimate parameter values in the population, then I look more towards making sure I fit each individual pretty well, right? And so, yeah, it depends on the application.
So do you have any optimum way of handling baselines for human data in these linear mixed effect models? That's one of the things so we use linear mixed effect models a lot for some of the clinical data, looking at the antibody responses, and the baseline variation always kills us. So I wonder if you have.
Yeah. No, I'm a believer in just estimating the initial condition. And so there are a lot of people who have had luck with feeding in the initial value, the first observation, as the initial condition, and might make the model more stable. But to me, it's putting the first observation on this pedestal that it's no more certain than any of the other observations usually, right? Unless they've done a triple baseline or something, in which case multiple baselines are nice when they're taken over multiple days, because it lets you get a sense of the natural history of the phenomenon that you're measuring. But normally, anyway, yeah, the fitting the baseline as an estimated parameter often works. Sometimes when your data is sparse and you have your baseline is 5 days before the start of treatment, and then your first measurement is 3 months after the start of treatment, there are many different upward trajectories a tumor could take and then come down to hit that point. And so sometimes it's very hard to observe that initial condition. Other times, depending on whether it's within weeks or just a couple of days of the start of treatment, other times it's very easy to estimate the baseline, the initial conditions. Is that what you were getting at?
Yeah, sounds good. I mean, yeah, that's similar thoughts.
Yeah. Some modelers take the baseline and just assume nothing happens between then and the start of treatment. That's also something I'm not crazy about, but it does let you get one less parameter sometimes to deal with. Yeah.
Depending on the sample size. So for the remaining group, I wonder what you think about doing the knowledge curation modeling groups. Should I try to push to get few people together, or I mean, I wonder the fate of that group.
Well, we'd certainly be delighted if you would push it. These are important topics.
Yeah.
And I wanted to say, just looking at your talk, it's hard enough when you do ODE models, where network models, dynamic network models, when you have unknown parameters. When you have actually structural unknowns in terms of the network itself, doing perturbation analysis on network structure is really challenging. And so to see you doing this formal analysis where you do a network perturbation, structural perturbations of the network was very interesting to me.
So Julie, are you talking about specifically management of or knowledge curation related to models, essentially a standard sort of like SPML or SPML, the discrete version of it, or are you talking about something more general?
So at least the working group was defined as knowledge acquisition and modeling subgroup. And I don't really know what it means to others, but for me, I was thinking actually simplification of literature-based or database information in the form of the networks. Doesn't have to be SBML. In any format that can be easily imported and translated. There are existing languages where I see the caveat is in actually gathering the information in the usable format. So I have seen like SBML mathematical models stored in SBML format, and but the translation of that from the existing information is not straightforward. So yeah, that's what I was thinking, at least for the type of knowledge acquisition that we are interested in thinking about, like for antiviral response, how can one create tools to gather or to create tools and standardize those to gather all the responses or molecular network associated with the antiviral response. So something like that. Those were the questions I was trying to address, or at least pose to this group.
So I think it's extremely important. You mentioned digital twins. If you don't have a good way of curating all the information, data, and models that go into that, it's a lost cause, right? Because it will become quite complex. So generally, I think this issue of knowledge representation and curation is extraordinarily important, and we have addressed that in part in some other contexts. And we want to discuss one of these, sort of basically creating a literature knowledge base tomorrow in the steering group meeting. If you can attend that, I think that'd be great.
Yeah, I will. I just moved my meeting to be able to attend that. So
perfect. That's great. Yeah. So we can certainly continue that discussion there.
Okay, great. Yeah, that will help. Yeah.
I mean, I think that feeds in very well with Dean's comment that model reproducibility is important, and that it's often, we run into this too. The software package we developed because 18 or 20 years ago, I tried to reproduce a paper that was published using a methodology that I invented, and I couldn't do it. And I thought that if I couldn't replicate a paper using the methodology that I developed, the probability that anyone else could replicate it was probably fairly small. And so we've spent 15, 20 years now trying to make models of the kind that we build more reproducible and more shareable, with mixed success, probably. And certainly things like SPML or CellML are trying to address some of these issues for the pathway modeling. But one of the things I think Reinhardt is alluding to that I'm always coming back to, people have heard me beat on this indefinitely, is that we don't have actually very good standards for describing conceptual models. Now you talked about biopacks and certainly specifically for biochemical networks, gene regulatory networks, there's some standards for how you represent the structural components of a model. But when you deal with higher level interactions, like immune response, which is not just a molecular set of interactions inside one cell, we don't really have a standard for how you describe what happens once you get outside of a single cell. And that inability to describe biological interactions in a portable way, I think is something that we're going to run into when we start trying to build more complex models that are usable and modular. And I don't know, Dean, I mean, again, if your focus is on building very simple models, then perhaps these are not issues that you run into so much. But if you think about, say, trying to build very elaborate physiological models with lots of components, being able to connect those components is not a trivial thing to do. And if you don't have the description of what the underlying biology was that led to that mathematics, it's not an easy thing to do. I mean, you show some Hill functions or some Michaelis functions. If those Michaelis functions are the result of, for example, your CD8 plus T cells take a certain amount of time to kill their target, and they have a certain velocity in the tissue, that's going to cause saturation of their ability to kill under certain circumstances. But the relationship between the cell velocity and the killing time and that saturation function isn't something very transparent, and you lose it when you write a simple functional relationship. So I wonder, both of you may have ideas about this issue of how do you represent the concepts in these scales.
Yeah, I remember longing for that, again, 10, 15 years ago in my, whenever I was at Physium, 20 years ago, for, an unambiguous physiological modeling language, basically. And yeah, like you said, CellML, SBML, CellML at least has like things like ion channels, right? But cell-cell interactions, they were working on tissue ML at the time, the same Auckland group. I don't know where that went, but yeah. So no, we're currently a partner, one of the sponsors of the 6 pharma company consortium for QSP models of a QSP platform of immune oncology, and so that's already an incredibly complicated network of we're in year 3 now, and hundreds of state variables, thousands of parameters, and every time you go through the literature, sometimes just to get the parameters, you end up reading another modeling paper. But again, the assumptions they put in there for the purpose that they were trying to model skews what that parameter value would be and whether it's useful for you. And it's very hard to evaluate that unless you know that if there was a common biological understanding map that we could all fall back on instead of each other's equation judgments. That'd be extremely helpful, I think.
So I wonder if the QSP consortium that you are talking about, do you then have the cell cytokine interaction network that is based on this? The models are based on the existing network or not?
Yeah, currently, you have certain cells that make certain cytokines and do certain things when they're exposed to other cytokines and so on and so forth. It's not an agent-based model, though. It's an ODE model, so the complexity gets very big. The more potential responses you could have, and if you have subpopulations of cells, then that starts getting out of control. Sort of like phosphorylation on a protein with 7 sites. It just gets out of control after a while.
So in this other world that I live in sometimes, so I think people are using the gene expression data to predict the cell cytokine interactions from I think there are some efforts from Broad. Is that what can be used in this case? And the other question I have, like, now the spatial transcriptomic data is becoming so popular, and there you actually know which cells are next to which cells, sort of, with the caveats again. But I mean, is this data then feeding into some of these models?
I don't think that data is. So the group leading it is a company called Certara, and they have a paper in CPT. I'm one of the authors on there. There's like 50 authors, so it's easy to find. P. van de Graaff is one of the other. Well, I don't have that many papers, so it's already easy to find. But and they allude to another method that they use RNA-seq data and then the immune landscape of cancer analysis that was done to the RNA-seq data from the, I think, the Cancer Genome Atlas, to determine relative abundances of cell types, a deconvolution type approach, right? Yeah. They use those deconvolved, which everyone in the biology world calls deconvoluted, which drives me crazy, but they use the deconvolved cell abundances to then prime the initial conditions in the consortium model. So that's one way they're using sort of omics type data. But the other mapping you talked about, I'm less familiar with. I don't know about, so I don't think they're using it there. Yeah. I think it's more like slogging through the literature about what cell types do when they're exposed to whatever, right? And they've defined a way. They've declared mouse to be out of scope, and so it kind of leaves you with ex vivo human experiments, human blood put on different assay conditions. Right. So I think that's most of it. Yeah. And they do have now, at our request, they had at first the cytokines were in the tumor microenvironment talking to each other. But then we pointed out all our trials generate tens of cytokines at many time points. And the holy grail is always, can you take a time course of a bunch of cytokines and infer not only what's going on in the tumor microenvironment, but is this patient now going to respond to this intervention favorably, or get a cytokine storm unfavorably, of course.
Yeah. It would be interesting. Like the spatial transcriptomic data now is.
I didn't catch the word patient. What data?
Spatial transcriptomic, the Visium technology.
Oh, transcriptomics. Okay. So
I think it's called. It's from 10x company, Visium. If not, I'm just writing.
There's also a milieu consortium. Have you all heard of that? Let me find the paper. I'll go on mute for a second while I'm trying to find this thing. I'll share the link.
Yeah, the only reason I was thinking about spatial transcriptomic data is because it limits what cells are exposed to what other cells. And not all the cells are going to see the cytokines that are expressed in the tissue environment. But again, I'm not from the oncology field. So.
Yeah, this is the one. I think this is the one. We've put it in the chat. So yeah, this is the example I mentioned before. I can share my screen if you want. Where's that little arrow? It's under my window. So this is not the paper. All right. Where is it? Okay. So yeah, Georgia Lazar, these are all, I think these are all authors from Certara. And yeah, just to drive home what I was talking about before. I think what they used was, now I'm wondering if this is the right paper. They used the TCGA, the Immune Landscape of Cancer Deconvolution efforts, to then map certain initial conditions and variability across the patient population for those initial conditions. So this is just a paper showing the examples of that. Now the one with immune milieu, I'm wondering if this is this paper or not. It's one of those, it's sort of one of those repository, but I don't know if it's omics data or I think it's just like an exhaustive library of what cells do when they're exposed to different things, kind of repository. So that might be helpful in the sort of building of knowledge around the immune system, knowledge maps and so on. But I don't know what their output looks like. I'm trying to remember if I ever actually looked into them besides the fact that they're referred to in either this paper or this other paper. I'm searching here. Paste this into the chat. No. No. Okay, let me just show you the paper. So speaking of maps, I thought this was a fascinating amount of work, especially they did an interdependency of immunology models map to help. So they did a very exhaustive literature search and then mapped which models were derived from which publication and so on. Anyway, that's a side issue thing. God, I can't figure out where I saw that immune milieu consortium. This event. Oh, this is a nice thing. Certara put together. So tumor cells release certain factors right here, which then stimulate or suppress right other cells. This is actually kind of a nice map. It is sometimes hard to take the dimensionality of biology and sort of map it in a visual way. But I thought this was a pretty good attempt here. So I want to see if the immune stuff was in there. Well, obviously that, but sorry, I have this diagram in my mind that I cannot find the source for, so I'll send it around later. Yeah, it's not here. Sorry. Okay.
That's okay. Thank you for looking. I've got that paper now, the link for that paper, and we'll. I'm sure Reinhardt and I will definitely look at it because that's very interesting to see somebody trying to put that kind of information together. Yeah. And clearly the immune, the immune response to viral infection, immune response to cancer aren't identical, but a lot of it is the same.
Yeah,
So-
and then that one resource I kept referring to and I can't find is yeah, based on viral perturbation, I guess, viral infection. Yeah, so no, the most of our sources are of an understanding of the immune system do come from viral infections, and they do seem to map pretty well to our understanding of tumor immunity, with some obvious differences, but yeah.
I mean, one thing that surprises me a little bit is just that I mean, one of the things we did is just taking the classic ODE models of infection and immune response, putting them into a spatial context. And now you're not in the stirred reactor anymore, and so diffusion matters, so distances matter. And one of the things you find, for example, is a simple parameter like beta, so that the probability that an uninfected cell becomes infected as a function of the viral load. That beta, where if you fit the ODE model to a set of experiment and pull that beta, the value of beta that you get is very different from the one when you include spatial effects.
Okay.
You can be off by a factor of 3 or 4 in terms of your beta estimate, because in fact viruses distribute locally, not
right.
And so you're underestimating your beta. A perfect fit of your ODE model will give you a beta that's too small, sometimes by quite a bit. And the same thing happens for all of the other cytokines because they take time to distribute in the tissue. Whether that's important or not, maybe it doesn't matter, because if you have everything aggregated, it may not matter. In other words, if you're modulating one particular factor using a drug, then that difference, it's not just it's a misfit rather than an error than noise. And so it's a systematic misfit that you get by assuming things are mixed.
Right. Yeah, it's something. It's a an assumption I had to sort of suck up, being kind of a spatial modeler in my academic career, that cells are points. That was the physiome paradigm, really. You build organs out of them, but intracellular spatial things. But no, you're absolutely right. And now we're getting, there's digital pathology is a thing, right? And so we are now trying to interpret patient biopsies and have more than just number of T cells and number of tumor cells in the same field, but what is the average distance and certain metrics on that. I always think it's tempting to just take some of these models that academic labs have been working on and sort of set the field that you see as the initial conditions and let them fight it out and see whether the tumor or the T cells win, right? But the big concern is always tumor heterogeneity. So you're getting a little tiny slice of a tumor, and it's hard to know how representative that is of what's going on in the patient tumor burden as a whole. So that's always the big challenge of spatial modeling and in clinical modeling is finding that right level of granularity. Yeah, there's some companies, what are they called? Anyway, they're doing CD8 imaging in vivo, and so once we have that and maybe a few other key markers that could be imaged simultaneously to a certain level of spatial resolution, then you'll see spatial modeling really picking up in immune oncology, I think. In clinical, in pharmaceutical modeling, that is.